Back to the schematic of the central nervous system structures, we are now ready to cover the next region of the CNS, which is the brainstem. Recall from our discussion about the CNS structures that the brainstem corresponds to the association between the midbrain, the pons, and the medulla oblongata. The brainstem is continuous with the spinal cord and the diencephalon, which, as you might remember, was composed out of the thalamus and the hypothalamus. Sometimes, parts of the diencephalon, such as the thalamus, are included in the brainstem, but for our purposes, we will exclude it and only cover anything that has to do with the diencephalon as part of the forebrain. Now, before we dive into the anatomy of the brainstem, we need to discuss what general roles and functions the brainstem mediates to get a general idea of the structure we will deal with. If you recall our structural and functional model of the nervous system, you will remember that in the most reductive form, the central nervous system, which contains the spinal cord, the brainstem, and the brain, are the intermediate between the perception of our senses and our motor actions. In our discussion about the spinal cord, we discussed at length how spinal nerves that leave the spinal cord carry afferent sensory information and efferent motor information. Additionally, we mentioned how the spinal cord has sensory motor reflexes and central pattern generator circuits that allow to do some processing of the environment. Nonetheless, much of the sensory information that the spinal cord received from the body left the spinal cord through ascending tracts that reached upstream structures like the brainstem and the brain. When it came to motor information coming from the descending tracts, these tracts originated in structures coming from the brainstem or the brain. For that reason, we can say that one of the functions of the brainstem is being a sensory motor intermediate between the brain and the spinal cord, as it contains pretty much every sensory or motor tract that the body uses. Across these tracts, some originate in the brainstem, some get modulated by the brainstem, and some send information to the brainstem for processing. So, although I call the brainstem an intermediate, a lot of processing happens there beyond simply relaying messages throughout the CNS. In addition to this intermediate role, the brainstem also directly receives sensory and sends motor information through structures called cranial nerves. As you can see on the schematic on the left, which is an interior view of the brainstem that we will cover shortly in more detail, cranial nerves are these yellow branches coming out of the brainstem, and they are somewhat similar to spinal nerves because they carry sensory motor information, but instead of supplying our limbs and trunk, cranial nerves supply the face and head region as well as our visceral organs. As a result, there are much more variety in the information they carry. There are 12 pairs of cranial nerves, and 10 of them come directly in and out of the brainstem. Accordingly, the brainstem is often regarded as being the key hub for these cranial nerves and associated nuclei. In terms of more miscellaneous processing, the brain has other nuclei scattered across the medulla, pons, and midbrain that deal with the processing of a wide array of information. To name a few, it contains nuclei responsible for modulating cardiovascular and respiratory activity, pain sensitivity, and other more abstract concepts like alertness, awareness, and consciousness. As you can see, the brainstem is a much more complicated structure in comparison to the spinal cord, as its structure is much less stereotypically organized, and due to its large amounts of different functional units that interact with the spinal cord and the brain, it mediates a lot of roles that are important for our survival. With this introductory overview of the brainstem functions, let's now focus on its external anatomy. To understand where the following diagrams come from, it will be important to recall the neuroanatomical navigation axes. The axis that will be important for us is the posterior dorsal and anterior ventral axis. So, if we first look at the brainstem from this perspective, we get the anterior view of the brainstem, and from this perspective, we get the posterior view. As you might see already, the external surface of the brainstem is much more different from the spinal cord, as it has many more bumps and ridges that will be important to consider. Another axis that will be important for us to keep in mind with the brainstem is the rostral and caudal axis. Remember that rostral is towards the nose and caudal is towards the tail. Accordingly, if we start from the most caudal end of the brainstem, we will get the junction between the spinal cord and the medulla oblongata. Moving rostrally, we obtain the pons, and more rostrally, the midbrain until we reach the diencephalon. Remember that in this video, we won't consider the diencephalon as a structure from the brainstem, and we will cover the details of this division like the thalamus and hypothalamus during our conversation about the brain. Now, in these two figures, there are some important anatomical landmarks that should be pointed out that will be helpful for reference later. First of all, you will notice on the caudal ends of both the anterior and posterior views the ventral and dorsal roots of the spinal nerves for segments C1 and C2, which mark the most rostral end of the spinal cord. Then, if we consider the interior view of the medulla, most medially we can observe the anterior median fissure that divides the pyramids of the medulla. As we will see later and in other videos regarding the motor system, the medullary pyramids carry descending motor information. In the pyramids, the region where the motor tracts cross is called the pyramidal decussation and represents an important landmark as it generally defines where the medulla and spinal cord divide. Moving laterally from the anterior median fissure, we have the anterolateral sulcus. This division separates the pyramids from the olives, which contain the olivary nuclei, an important cluster of neurons that we will cover later. At the most rostral end of the medulla, there is the medullopontine sulcus that divides the medulla from the pons. Moving rostrally in the brainstem, another prominent structure to note are the cerebral peduncles that are most notably known for carrying descending motor information. In the most medial region of the midbrain diencephalon region, there are two important structures that are characteristic of the interior view: the mammillary body and the pituitary gland that is connected to the hypothalamus by the infundibulum or pituitary stalk. These two structures, along with the thalamus that you can see more rostrally, are part of the diencephalon, so we will see in more detail their roles later. Now, when it comes to the posterior view, in the medulla it is important to point out the posterior median sulcus that medially separates the two sides. More rostrally in the pons, an important structure is the cerebellar peduncles that connect the brainstem, brain, and spinal cord with the cerebellum. More rostrally in the midbrain, two of the most characteristic structures of the posterior view are the inferior and superior colliculi, which respectively help with the processing of auditory and visual information. Then, rostral to the superior colliculi, we have the pineal body, which is an important structure of the hypothalamus. Now, although for all of these structures that I've pointed out, I will cover their roles later or in other videos, it's good to see their overall shapes and where they are now, because they will act as good reference points when we will consider cross-sections of the brainstem. Finally, the last and maybe one of the most important anatomical detail to consider with respect to the brainstem are the multiple cranial nerves in yellow you see coming out of it, which we are now ready to consider in more detail. If you remember our discussion on the different roles and functions of the brainstem, you will recall that there are 12 pairs of cranial nerves and 10 of them come directly from the brainstem. Now, two important things you will notice with these diagrams is that first, the interior view on the left is much more informative about the cranial nerves in comparison to the posterior view. For that reason, while discussing the cranial nerves, we will mostly consider the interior view. The second thing that you will notice is that from these two diagrams, we can only see 11 out of the 12 cranial nerve pairs. To see the full 12 pairs, we need to consider this ventral or bottom view of the interior surface of the brain. When we compare this figure with the interior view we have been using, I hope that you can see the brainstem in more context and how it's structured with respect to the brain. So, as I said, 10 of the 12 pairs are related to the brainstem, which leaves 2 pairs out of it. These 2 pairs correspond to cranial nerve 1, the olfactory nerve, and cranial nerve 2, the optic nerve. By the way, before we get into it, I want to mention that the 12 pairs are numbered 1 to 12 based on a rostral to caudal direction, meaning that the first cranial nerve will come from the brain and the 12th one will be at the most caudal end of the medulla, so when we will go through them, we will roughly go from a rostral to caudal direction. Also, I want to mention that for any cranial nerve, it is widely accepted to either call them directly by their names or by their cranial nerve number, which is often noted in Roman numeral form, but I personally find it easier to remember them with regular numbers. Finally, you will often see the abbreviation CN, like here for CN1 and CN2, and it simply means cranial nerve 1 and 2. Alright, so back to the olfactory and optic nerves, the two convey purely sensory information to regions out of the brainstem. Starting with the olfactory nerve, we can best appreciate its function through this mid-sagittal diagram of the human face. To get oriented, here is the tongue and hard palate that illustrate the position of the mouth. Superior to the hard palate, we have the nasal cavity where the olfactory nerve plays an important role to mediate our perception of smell. Indeed, the nasal cavity contains the olfactory epithelium, which contains the cell bodies of olfactory receptor neurons that transduce odorant chemicals into electrical signals. These electrical signals then pass through the cribriform plate via the many olfactory nerves to reach the olfactory bulb. After some processing in the bulb, the signals are sent to different areas of the brain through the olfactory tract. In all, the function of olfactory nerves can pretty much be summarized to them being the carriers of afferent information about olfaction from the olfactory epithelium to the brain. Now, when it comes to the optic nerve, it carries afferent information about vision from the retina to the brain. If you are not familiar with the retina is a layer of cells at the back of your eye, and it contains photoreceptors as well as supporting cells that altogether convert visual information to neural signals. These signals are then funneled into the optic nerve, which connects the eye to the rest of the brain. Obviously, the broader context behind these cranial nerves and some additional information about these diagrams will be explained in more detail in their respective videos when we will cover sensory systems. But for now, this should suffice us for us to understand their roles. Very simply, the olfactory nerves carry sensory information for the sense of smell, and the optic nerve carries sensory information for the sense of vision. So, with these first two cranial nerves covered, we can return and focus on the 10 remaining cranial nerves that come out of the brainstem. But before we go through them one by one to get a general feel for their roles, I want to first show you an overview of all these cranial nerves to point out some aspects that will be important to keep in mind throughout our discussion. The way I have decided to organize this overview is to separate the cranial nerves based on the region they leave from in the anterior view. Accordingly, the first region is outside of the brainstem, which contains the first two cranial nerves we have covered. Then we move into the midbrain, where cranial nerves 3 and 4 exit. Technically, cranial nerve 3 exits at the midbrain Pons junction, but to say it leaves at the midbrain is good enough for us. In the Pons, the only cranial nerve to exit at this level is cranial nerve 5, and at the pontine medulla junction, there are 3 nerves from cranial nerve 6 to 8 that leave. Finally, the 4 remaining nerves exit from the medulla. The first important aspect that I want you to notice is the fact that cranial nerves can either carry sensory information like the first two we have seen, but they can also carry motor information, or in some instances, they can be mixed and carry both. This directly contrasts what we have seen with spinal nerves, as they were all mixed nerves that carry both sensory and motor information. Now, although some nerves are very specialized for one specific task, like cranial nerves 1, 2, 4, 6, 8, and 12, some mediate a much more diverse set of functions, especially 7, 9, and 10. Accordingly, a given cranial nerve can contain multiple different fibers that go on to operate their respective functions. Depending on where these fibers go or come from, they can be categorized in 7 different modalities that describe what type of information is being transmitted. The 7 different modalities are represented by the 3-letter abbreviations you see before each function. To understand what these abbreviations mean, recall that, generally speaking, the fibers entering the central nervous system, or in this case the brainstem, are called afferent fibers and carry sensory information. On the other hand, the fibers that emerge from the brainstem are called efferent fibers and carry motor or autonomic information. Both the afferent and efferent fibers can then further be divided into 7 different types of fibers, with 4 being sensory and 3 being motor. Now, you will notice that for all of these 7 fiber types, the first word will either be general or special, and the second word will either be somatic or visceral. If we start with the second word first, the difference between somatic and visceral is that somatic relates to the body and more precisely to the skin, muscles, and joints, whereas the visceral relates to the internal organs. As a result, all the fibers that go, let's say, to the heart or the lungs will be classified as visceral because they go to internal organs, but any fibers that goes to the skin will be labeled as somatic. For general and special fibers, let's start with sensory fibers first. In this instance, general afferent fibers are the ones that carry information about touch, pain, temperature, and proprioception, which primarily comes from the skin, muscles, and joints, whereas special fibers carry information from our special senses like vision, hearing, balance, olfaction, and taste. In terms of efferent fibers, general efferent fibers send information to skeletal, smooth, and cardiac muscles, as well as glands. Special efferent fibers, on the other hand, activate muscles that are derived from pharyngeal arches, which are basically the muscles that constitute the face, the pharynx, the larynx, and a few other structures in the head. Altogether, the main difference between general and special fibers is whether or not spinal nerves are also capable of carrying the information. For example, the senses coming from our head like vision, olfaction, and so on are only carried by cranial nerves, but our senses of touch, pain, and proprioception are both carried by cranial and spinal nerves, which is why only the fibers carrying vision and so on are said to be special, and the same logic goes for efferent fibers. In summary, all 7 distinctions of fibers can be summarized by these 3 pairs of words, with the first being either special or general, the second being either somatic or visceral, and the third being either afferent or efferent. Accordingly, as long as you know the difference between each word and what regions they correspond to, it should be easier to derive the different types of fibers. The only difference I would like to explicitly point out is between special visceral afferents and special somatic afferents. As you can see, SVA fibers in green are responsible for olfaction and taste, whereas SSA fibers in blue are responsible for vision, audition, and balance. The key difference between them is the somatic and visceral distinction, and that is explained by the fact that the senses supplied by the SSA fibers are derived from ectoderm, whereas the senses supplied by SVA fibers are derived from endoderm. With the exception of this distinction between these 2 types of fibers, the rest should be straightforward to determine. Now, although I have decided to organize the cranial nerves based on where they exit in the brainstem, it can also be good practice to reorganize the cranial nerves based on their modalities to understand how they compare to one another. This can for sure help study their functions and maybe highlight commonalities or differences that are not apparent under this view. The final aspect that I want to discuss before we jump into specifics of the cranial nerves concerns what actually happens inside the brainstem. Indeed, although the external surface of the interior view we have been using shows us the nerves that leave the brainstem, it is also important for us to consider the nuclei that the nerves target or originate from. To do so, we will consider the posterior view, in which we can see the many nuclei that fill the brainstem. Remember that a nucleus is defined in the central nervous system as a cluster of neuronal cell bodies that usually share some kind of feature. The 2 broad distinctions for the nuclei in the brainstem are sensory and motor. In this figure, the sensory nuclei are located on the left and the motor nuclei are located on the right. All of these nuclei are obviously bilateral but are shown on one side for simplicity. Broadly speaking, sensory nuclei are the targets for afferent cranial nerves, and motor nuclei hold the cell bodies of the neurons that constitute efferent cranial nerves, but it happens in some instances where the nuclei are somewhat mixed. In terms of their disposition, sensory nuclei are generally found laterally, whereas motor nuclei tend to be more medial. An important aspect between cranial nerve and nuclei, not to be confused about, is that there is not a one-to-one correspondence between nerves and nuclei. For example, the trochlear nucleus entirely supplies the trochlear nerve, but in other instances, there are nuclei such as the nucleus solitarius that integrates information from many cranial nerves. Accordingly, it will be very important to take the different nuclei of the cranial nerves into account to get an appreciation of their function. With this overview now established, we can begin our discussion about the cranial nerves in the brainstem, starting with cranial nerve 3, the oculomotor nerve, cranial nerve 4, the trochlear nerve, and cranial nerve 6, the abducens nerve. These 3 nerves generally fall under the umbrella of oculomotor nerves, as they are responsible for the movements of the eye. To discuss these 3 cranial nerves, let's first take 3 cross-sections of the brainstem to see where their nuclei reside, because it turns out that each nerve has its respective nucleus. Starting with the oculomotor nerve, we can find the oculomotor nucleus at the level of the superior colliculus in the midbrain. A little more caudally, at the level of the inferior colliculus, we can find the trochlear nucleus, and finally, we can find the abducens nucleus. Since all of these nuclei supply motor nerves only, they house the cell bodies of the neurons that will send projections that will exit the brainstem. One particularity you will notice with the projections coming from the trochlear nucleus is that first, they cross and they go to the opposite side from where they originate from, while the projections from the oculomotor and abducens nucleus stay on the same side. This means that the projections from the left trochlear nucleus will reach the right eye, and the projections from the right trochlear nucleus will reach the left eye. Secondly, the projections from the trochlear nucleus leave the brainstem from the posterior side. This aspect is very characteristic of the trochlear nerve, as it is the only cranial nerve leaving from the posterior side. Now, in terms of their function, as I said, they are responsible for the movements of the eye, so let's consider the muscles that generate the movements. It turns out that all of the possible motions of our eyes are mediated by 6 different muscles: the superior, medial, inferior, and lateral recti, as well as the superior and inferior obliques. The superior oblique is a bit different from the other muscles, as it contracts the eye through a small tendon called the trochlea. When we consider what cranial nerves innervate what muscles, cranial nerve 4 and cranial nerve 6 are each responsible for one of the 6 eye muscles. As you might guess, due to the trochlear nerve innervates the superior oblique. On the other hand, the abducens nerve innervates the lateral rectus, and the 4 remaining muscles are innervated by cranial nerve 3, the oculomotor nerve. Since all these muscles are striated, the fibers that innervate the muscles are labeled as general somatic efferent, or simply GSE. Now, as far as this video goes, we won't discuss the process of eye movement, but note that all 3 cranial nerves interact with each other in some antagonistic way to produce fluid eye movements. To do so, the 3 nuclei communicate with each other to coordinate the movements, and this is done through the medial longitudinal fasciculus, which is a white matter tract that integrates their signals with other brain centers to mediate fluid eye movements. Beyond controlling the muscles that move the eye, the oculomotor nerve has other functions that are important to consider, which we can observe best with a lateral view of the eye. You will recognize with the orange and blue colors the respective muscles each cranial nerve innervates. Along with the muscles, the lateral view also includes the oculomotor nerve, which divides into a superior and inferior branch when it reaches the region of the eye. The inferior branch innervates the inferior rectus, the inferior oblique, and the medial rectus. The superior branch innervates the superior rectus and the levator palpebrae superioris. The levator palpebrae superioris is a muscle outside of the 6 that control eye movements, and it is responsible for raising the eyelid when it is contracted. To understand the final function of the oculomotor nerve, it is important to understand that the fibers that travel inside of the oculomotor nerve do not only originate from the oculomotor nucleus, as it turns out that the oculomotor nerve also carries fibers that originate in the Edinger-Westphal nucleus. The Edinger-Westphal nucleus gives rise to the preganglionic parasympathetic fibers that travel through the oculomotor nerve to reach a structure called the ciliary ganglion, which lies close to the optic nerve, or as we've seen, cranial nerve 2. By the way, remember that in the peripheral nervous system, ganglions are defined as a collection of neuronal cell bodies. Now, if we take a closer look at the eye, the ciliary ganglion contains postsynaptic fibers that, when activated by the presynaptic fibers of the Edinger-Westphal nucleus, will activate the ciliary muscle to constrict the pupil. The constriction of the pupil will have the effect of reducing the amount of light entering the eye, and this is an important effect of the parasympathetic response. Additionally, the contraction of the ciliary muscle can change the shape of the lens, and that allows for the ability of our eyes to focus light from objects at varying distance on our retina. This mechanism is also known as accommodation. In all, this packed diagram should summarize the main functions and ideas behind cranial nerves 3, 4, and 6. With these 3 cranial nerves covered, we can move on to the next cranial nerve, the trigeminal nerve. In comparison to the other cranial nerves, the trigeminal nerve is very special because its respective nucleus is located all over the brainstem, as you can see in green. In total, there are 4 divisions of the trigeminal nucleus, and to see them more clearly, let's take a cross-section at the level of the midbrain, pons, and medulla. Starting at the pons, we can find 2 divisions, namely the principal sensory nucleus in orange and the motor nucleus in red. In terms of their functions, the principal sensory nucleus mostly receives sensory information about touch from the face and proprioception from the jaw. On the other hand, the motor nucleus is responsible for innervating the muscles of the first pharyngeal arch, which are mainly the muscles of mastication, and the tensor tympani, a small muscle in the ear. The motor nucleus also will send projections to innervate other muscles such as the tensor villi palatini, the mylohyoid, and the anterior belly of the digastric, but the mastication muscles and the tensor tympani are the most relevant for our purposes. If we go at the medulla level, we can find the spinal nucleus, which is an important sensory nucleus that receives information about touch, pain, temperature, and proprioception for the entire face. Finally, at the level of the midbrain, we can find the mesencephalic nucleus that processes mainly proprioceptive signals from mastication, neck, and head muscles. In all, out of the 4 nuclei, 3 are sensory and 1 is motor. Now, an important characteristic of the trigeminal nerve is that it turns out that it is actually the largest cranial nerve out of them all. This is in part caused by the fact that the sensory signals coming from the face that innervate the trigeminal nuclei come from the trigeminal ganglion, which is best appreciated on the lateral surface of the face. The trigeminal ganglion is to some extent analogous to the dorsal root ganglion that we have covered in our discussion about the spinal cord, as it contains the cell bodies of the pseudo-unipolar neurons that will reach each sensory nuclei. You will notice that the trigeminal ganglion has 3 different branches and each innervate a specific region. If you remember again from our discussion on spinal nerves, these specific regions are called dermatomes and essentially correspond to the regions of the body that are supplied by specific divisions of nerves. The first and most superior branch of the trigeminal ganglion is called the ophthalmic nerve and supplies the upper portion of the head. As a shorthand, this division is abbreviated to V1. As you might imagine, the next division is labeled V2 and it corresponds to the maxillary nerve. Finally, the last division, V3, is the mandibular nerve. With these 3 dermatomes, we can complete our picture about the dermatomes for the entire body when we consider the dermatomes from the cervical segments that innervate the neck and the back of the head. As a final note, I want to mention that the projections from the motor nucleus travel exclusively through the mandibular nerve division, or V3, to reach their targets. This last detail completes our picture about the trigeminal nerve, which means we are ready to move on to the next cranial nerve, the facial nerve. To cover its functions, let's take 2 cross-sections, one at the level of the pons and one at the level of the medulla. In the pons, we can first find the motor nucleus of the facial nerve, which sends projections to innervate the muscles of the second pharyngeal arch, and these correspond mostly to the muscles responsible for facial expressions and the stapedius, a small muscle in the ear. There are other muscles in the second pharyngeal arch, such as the posterior belly of digastric and the stylohyoid, but for our purposes, the muscles of facial expressions and the stapedius are the most relevant. In the pons again, there is a second nucleus that produces fibers for the facial nerve. It is called the superior salivatory nucleus. The superior salivatory nucleus is responsible for sending preganglionic parasympathetic fibers to 2 ganglions that then relay the signal on important glands of the face to make them secrete their respective fluids. The first ganglion is called the pterygopalatine ganglion, and it connects the lacrimal and mucosal glands of the oral and nasal cavities. The lacrimal glands are located near the eyes and produce lacrimal fluid, more commonly known as tears. The mucosal glands can be found in the sinuses of the face and produce mucus. The second ganglion that is connected to the superior salivatory nucleus is the submandibular ganglion, and its activation activates the submandibular and sublingual salivary glands. As you might suspect, these glands produce saliva. I should also mention that there is a third salivary gland called the parotid gland, but this one gets activated by cranial nerve 9. Now, when it comes to the medulla, it contains an important sensory nuclei for the facial nerve, namely the nucleus solitarius. This nucleus receives taste sensations of the anterior 2/3 of the tongue from the facial nerve. The final detail that I want to mention with respect to the facial nerve is that it also conducts sensory fibers. If you recall the spinal nucleus of the trigeminal nerve, you will remember that its main function was to be a receptive zone to the touch, pain, temperature, and proprioceptive sensations of the face. It turns out that the facial nerve feeds this receptive zone by carrying GSA fibers that innervate small structures in the ear, such as the tympanic membrane, the external auditory meatus, and a small portion of the auricle. Although this detail might not be the most memorable feature of the facial nerve, it is good to keep in mind that cranial nerve 7 also supplies some sensory sensations in the ear. So, with this detail now covered, we have went through most of the essentials of the facial nerve. We can now shift our attention on cranial nerve 8, the vestibulocochlear nerve. So, in comparison to the other cranial nerves found in the brainstem, the vestibulocochlear nerve is rather simple because it is primarily sensory and it innervates mostly one structure, the inner ear. To find that inner ear, we need to first consider the general anatomy of the ear. The ear is generally divided into three sections: the outer ear, the middle ear, and the inner ear. Broadly speaking, the general function of the outer and middle ear is to funnel sounds on the inner ear. Now, the inner ear itself is divided into two principal functional units. First, there is the snail-shaped structure called the cochlea, which, when we consider its cross-section, contains specialized cells that transduce the vibrations from sounds into neural signals. These neural signals are then picked up by the cochlear division of the vestibulocochlear nerve and carried to the central nervous system to create our auditory perception. The second functional unit of the inner ear is the vestibular system, which is generally composed out of the semicircular canals and the otolith organs. These structures contain specialized cells as well, but do not process sounds. Rather, the specialized cells in the vestibular system sense our motion to produce our sense of balance. The signals picked up from the vestibular system in the inner ear are carried to the CNS via the vestibular division of cranial nerve 8. Altogether, the cochlear and vestibular divisions travel together to reach the pontine medullar junction to synapse on their respective nuclei. Of course, there is much more information to be said about the anatomy and physiology of the ear, which we will do in due time when we will cover the auditory and vestibular systems. One detail, however, that I wish to mention before we move on concerns the two muscles you might see in the middle ear, the stapedius and the tensor tympani. If you recall our discussion about the trigeminal and facial nerves, you might remember these names as they were the muscles that the respective motor nuclei from cranial nerve 5 and 7 innervated. With respect to their function, these muscles are very important because they contact tiny bones in the middle ear that conduct the sound. So, when these muscles contract, they dampen the intensity with which the sounds are transmitted to the inner ear. As you can imagine, any deficit with these muscles can lead to very severe conditions because the sounds will be perceived as much louder and might cause great amount of pain. Alright, now the next cranial nerve we will go over is the glossopharyngeal nerve, which, as you can see, has many different fibers that mediate a bunch of different roles in the body. Accordingly, the glossopharyngeal nerve is supplied by many nuclei in the brainstem. Fortunately for us, we can all find them at roughly the same cross-section in the rostral medulla. The first important nucleus that provides fibers to the glossopharyngeal nerve is the nucleus ambiguus. This nucleus provides the origin for the motor neurons that will innervate the stylopharyngeus, which is the only muscle part of the muscles derived from the third pharyngeal arch. When the stylopharyngeus contracts, it allows to elevate the pharynx and the larynx, which in return facilitates the process of swallowing. The second nucleus that provides fibers to the glossopharyngeal nerve is the inferior salivatory nucleus. This nucleus provides preganglionic parasympathetic fibers to the optic ganglion, which then sends postganglionic fibers onto the parotid gland. Recall from our discussion on the facial nerve that the parotid gland was the third salivary gland and that the two other glands, the sublingual and submandibular glands, were activated by the facial nerve. The third important nucleus is the nucleus solitarius, which primarily receives sensory inputs from the glossopharyngeal nerves. Some of these fibers are visceral afferent fibers and bring taste sensations of the posterior one-third of the tongue. Remember that the remaining taste sensations from the anterior two-thirds were carried by the facial nerve. In addition to these fibers, the glossopharyngeal nerve also carries general visceral afferents that innervate the carotid body and the carotid sinus. To find these two structures, we can consider how the heart provides oxygenated blood to the head and the neck. The main blood supply originates in the common carotid artery and then branches into the internal and external carotid arteries. At the bifurcation of the internal and external carotid arteries is where both the carotid sinus and body are located. Starting with the carotid sinus, it is a dilation at the base of the internal carotid artery that contains specialized cells called baroreceptors, which have the primary function of sensing blood pressure. Still, at the bifurcation, we can also find the carotid body. The structure is essentially a cluster of specialized cells called peripheral chemoreceptors. These peripheral chemoreceptors have the important role of sensing the blood's partial pressure of oxygen and carbon dioxide, as well as the blood's pH. The final important nucleus of the glossopharyngeal nerve is the spinal nucleus of the trigeminal nerve. Again, recall that this nucleus receives all kinds of somatic afferent information such as touch, pain, temperature, and proprioception from the face. It turns out that the glossopharyngeal nerve also feeds information into this nucleus by supplying somatic information about some structures in the ear, tongue, and upper pharynx. Given that the pharynx is considered to be part of the viscera, these specific fibers going to innervate the structures of the upper pharynx are considered to be general visceral afferent. This final detail completes our general picture of the glossopharyngeal nerve. With the glossopharyngeal nerve now covered, we can move on to cranial nerve 10, the vagus nerve. The vagus nerve is in some way a bit similar to the glossopharyngeal nerve, as all of the nuclei from where the different fibers originate or terminate can be found in the same cross-section in the rostral medulla. Also, much like the glossopharyngeal nerve, the vagus nerve is a mixed nerve carrying both afferent and efferent signals, but the vagus nerve differs from others as it is the only one that leaves the head and neck region of the body. As a result, it is important to note that the vagus nerve is the longest cranial nerve. This aspect can be appreciated from the first important nuclei, the dorsal nucleus of vagus. This nucleus provides parasympathetic innervation to the gastrointestinal tract, the lungs, and the heart. To be more precise, the more extensive list of structures that it supplies are the following, which you can consider if you need to. Remember that the main purpose of the parasympathetic nervous system is to promote digestion and slow down movement to conserve energy. It turns out that the vagus nerve carries the vast majority of all parasympathetic fibers, and as such, its main function is often associated with these fibers from the dorsal nucleus of vagus. One detail I'd like to point out is that this parasympathetic supply from the vagus nerve stops at the splenic flexure of the large intestine. Now, the second important nucleus is the nucleus ambiguus, and its most notable function through the vagus nerve is providing special visceral efferent fibers that will innervate muscles derived from the 4th and 6th pharyngeal arches. Broadly speaking, there are some exceptions, but the muscles of the 4th and 6th pharyngeal arches are basically the constrictor muscles of the pharynx, muscles of the soft palate, and some intrinsic muscles of the larynx. To get a quick appreciation of what these muscles mediate, we can consider this lateral view where we can see the pharyngeal constrictor muscles being attached to the larynx. When they contract, these muscles allow to constrict the esophagus, and that results in facilitating the process of swallowing, also known as deglutition. As for the intrinsic muscles of the larynx, their contraction contributes to the production of speech, or in other words, phonation. The second important role of the nucleus ambiguus is to send preganglionic parasympathetic fibers to the heart, and the downstream effect of that will be to decrease the heart rate. The next nucleus that has important implications for the vagus nerve is the nucleus solitarius, which receives many different sensory signals from the vagus nerve. The first important sensory signal comes from the taste receptors located in the epiglottis and pharyngeal region that transmit taste. Recall that taste from the tongue was carried out to the nucleus solitarius via cranial nerves 7 and 9. The second sensory signal that the vagus nerve carries to the nucleus solitarius is homeostatic information from the aortic bodies and baroreceptors. These structures in question are located at the aortic arch, which is the portion of the aorta that leaves the heart. Much like the carotid body and sinus, the baroreceptors and chemoreceptors that the vagus nerve innervates respectively signals information about blood pressure and about the partial pressures of oxygen and carbon dioxide in the blood and its pH. In addition to these sensory signals, the nucleus solitarius also receives visceral sensations with the exception of pain from the internal organs. Now, the final nucleus that is important to the vagus nerve is the spinal nucleus of the trigeminal nerve, which here again integrates information about touch, pain, temperature, and proprioception for the entire face. The vagus nerve brings to this nucleus general sensory afferent information from the outer ear, the tympanic membrane, the dura of the posterior cranial fossa, which is the cranial cavity at the base of the skull, and from the mucosa of the larynx. Again, since the larynx is part of the viscera, these sensory fibers are labeled as general visceral afferents. As you can see from this diagram right now, the vagus nerve mediates a ton of functions, and accordingly so, there is a ton of information displayed currently. So, make sure to go through it slowly if it's the first time you see these terms. Alright, We now have the last two cranial nerves, cranial nerve 11 and 12, to cover. Since they are rather simple as they mediate mostly one function, we will cover them together. Starting with the accessory nerve first, it is an interesting nerve with respect to the others because it contains both a spinal and a cranial division. As you might imagine, to find the spinal division, we need to consider a cross-section of the spinal cord to find the spinal accessory nucleus in the lateral horns. This nucleus is essentially a row of motor neurons that span from cervical segment C1 to C5. In contrast to the motor neurons we have seen that exit the spinal cord through the ventral root, the motor neurons in the spinal accessory nucleus exit through the spinal accessory nerve, which lies between the ventral and dorsal roots, to innervate two distinct muscles, the sternocleidomastoid and the trapezius. When it contracts, the sternocleidomastoid muscle has the function of rotating the head and flexing the neck. When the trapezius contracts, its main function is to move the scapula, also known as the shoulder blade. Now, the spinal accessory nucleus obviously ends when the brainstem begins, but it is believed that this nucleus is continuous with the nucleus ambiguus in the medulla, and that gives rise to the cranial division of the accessory nucleus. The cranial division of the accessory nerve, coming from the nucleus ambiguus, runs in parallel with the spinal division for a brief period of time, but then shortly after, it separates to innervate some muscles of the soft palate, pharynx, and larynx with cranial nerves 9 and 10. This grouping of nerves, including the cranial nerve 9, 10, and the cranial division of cranial nerve 11, is also known as the pharyngeal plexus. Now, because the core function of the cranial division is somewhat also attributable to the vagus nerve, the accessory nerve is most memorable for its spinal division that activates the sternocleidomastoid and trapezius muscles. Accordingly, if you see the accessory nerve being called the spinal accessory nerve, it simply means that the person behind has chosen to only consider the function of the spinal division. Now, finally, we can move on to the last and 12th cranial nerve, the hypoglossal nerve. Very simply, the fibers of the hypoglossal nerve originate in the hypoglossal nucleus in the medulla, which can be found at the same level as the nucleus ambiguus. This nucleus gives rise to the motor neurons that go on to innervate the intrinsic and extrinsic muscles of the tongue. The only muscle of the tongue not to be innervated by the hypoglossal nerve is the palatoglossus. This final detail completes our discussion on each cranial nerve. Back to this overview, with the information we have accumulated about these cranial nerves, we can now better appreciate the diversity and function that each nerve mediates. We can also appreciate the fact that, for the most part, the fibers that make up these cranial nerves target or originate from a bunch of nuclei in the brainstem, and that makes the brainstem a very complex structure that integrates an immense array of functions. With this being said, however, there is much more about the brainstem we have not covered in our conversation about cranial nerves. So, before we move on to the brain, I want to take a moment to briefly point out some key structures in the brainstem. Obviously, I will not go in much detail about these structures as, for one, if I did it, the video would never end, but most importantly, we will revisit most of these important structures in their respective context. Now, to see some of these structures, let's begin at the medulla, where we will take 3 cross-sections, one at the caudal end of the medulla, one at the middle level of the medulla, and one at the rostral end of the medulla. To get you situated on the anterior-posterior or ventral-dorsal axis, the side pointing towards the bottom is the ventral or anterior side, and the side pointing up is the dorsal or posterior side. This orientation holds for all of the cross-sections of the brainstem and spinal cord you will see in this video. Now, if you recall our discussion on the spinal cord, you will remember that the 2 key components of any cross-section in the nervous system are the gray and white matter. For the most part, gray matter corresponds to the cell bodies of neurons, and white matter corresponds to the axonal projections that are enwrapped in myelin. In the spinal cord, the gray matter had this butterfly or capital H shape and was located in the middle of the cross-section. We further divided the gray matter into 10 divisions called rex laminae that each operated a particular task across the spinal cord. When it comes to the brainstem, instead of talking about rex laminae, you have seen throughout our discussion on cranial nerves that the gray matter is organized in different nuclei that are scattered all around the brainstem. In the medulla, the 2 main different types of nuclei that we can observe are sensory and motor nuclei. For the most part, these are all nuclei that we have seen across the cranial nerves profiles. One aspect of the terminology about the gray matter that I'd like to point out, which you might see sometimes in your textbooks, is the concept of the reticular formation. Simply put, the reticular formation represents the central core of nuclei and gray matter of the brainstem. A bit like the butterfly shape in the spinal cord, but considerably more complex due to the different neuronal circuits it contains. For the white matter, the spinal cord was divided in primarily descending or ascending tracts. As a general rule, ascending tracts gathered information from the periphery and carried sensory information to reach targets located in the brainstem, the thalamus, or the cerebellum. Inversely, descending tracts originated in the brain, brainstem, or cerebellum and carried motor information that would eventually reach the periphery. As a result, when we consider the medulla, we will come across a lot of these ascending and descending tracts that carry information to their respective targets. In addition to them, the medulla also includes mixed tracts such as the inferior cerebellopeduncle and the medial longitudinal fasciculus that carries information in both directions. As I said, we will not go through all of these tracts and nuclei in detail until we encounter them in their context, but feel free to use these diagrams to get an idea of what's in the medulla. Of course, I want to point out that these diagrams are rarely anatomically accurate, as, to be honest, the real cross-sections of the brainstem are very messy and hard to anatomically distinguish, which makes every idealized cross-section diagram like this be different. As such, if this diagram differs from your textbook, it's perfectly normal, as every textbook from which I have integrated this information disagrees in some way with one another. Nonetheless, moving on rostrally to the pons, we will consider two cross-sections, one at the caudal level and one more at the middle level of the pons. Here again, just like in the medulla, the pons contain different sets of gray and white matter. The gray matter nuclei can be broadly divided into sensory and motor nuclei, and the white matter that crosses the pons can be divided into ascending, descending, and mixed tracts. In terms of the structure and how things operate, the pons are to some extent similar to the medulla as they contain the same broad types of divisions. Now, where it gets a bit more interesting is at the level of the midbrain. Here, we will consider two cross-sections, one at the level of the inferior colliculus and one more rostrally at the level of the superior colliculus. Before we get into the structures that compose the midbrain, there is one recurrent anatomical aspect that I want to point out. The midbrain is generally divided into three distinct sections. The most dorsal or posterior section is called the tectum, and it corresponds to the combined regions of the inferior and superior colliculi. The most ventral section is called the cerebral peduncles, and the region in between the tectum and the cerebral peduncles is called the tegmentum. Now, at this level of the brainstem, we can find sensory and motor nuclei, as well as ascending, descending, and mixed tracts, but one interesting type of structure that we can start to observe are nuclei that I will label as mixed nuclei. The reason why I call these nuclei mixed is mostly because their projections mediate a ton of different functions that are kind of hard to classify as mostly sensory or motor. If you recall in one of our discussions about neurotransmitters, where we covered the different catecholamines like dopamine, norepinephrine, serotonin, and so on, you might remember that the origin of the neurons that release specific neurotransmitters were localized in some specific regions in the brainstem. For example, we can first consider the locus coeruleus, which is an important nuclei that contains cell bodies of the neurons that release norepinephrine. As we mentioned in the discussion about norepinephrine, the projections coming out of this nucleus go all over the brain and influence different components of our lives, such as our sleep, levels of wakefulness, arousal, and attention. Additionally, it is also known that the locus coeruleus is involved in pain modulation. Another example of this multi-integrative nuclei is the raphe nuclei, which predominantly contains neurons that release serotonin across the central nervous system and influence our emotions, our hunger, our motor movements, and many more. Some other examples include the substantia nigra, which notably releases dopamine and influences our movements and our perception of reward. Again, these are all structures that for the most part, we will encounter them again in upcoming videos about different sensory and motor systems. With the midbrain covered, we can now get a good overview of the anatomy of the brainstem that we went through. We started our discussion by describing the different functions of the brainstem, and then we covered its external anatomy. Then, we moved on to cover the principles behind the cranial nerves, and then we jumped into their individual profiles from cranial nerve 1 to cranial nerve 12. In these discussions, we mostly focused on the associated nuclei and their respective functions without bothering too much about their courses in the body. Finally, we discussed the gray and white matter divisions in the cross-sections of the medulla, pons, and midbrain. This concludes our discussion on the brainstem, and this means we are ready to move on to the next portion of the central nervous system. Based on the embryological divisions, the next section that we will cover corresponds to the prosencephalon or forebrain here in blue. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. On the right, you will see the informational resources that I've used to produce this video. Thank you again for watching, and I'll see you in our next discussion.